Assalamu alaikum. Today I am going to discuss about enzyme. How enzyme defined in medical biochemistry. So enzymes are the biological protein catalyst, colloidal and heat level produced by living cells catalyze the biochemical reaction and remain unchanged in overall process. So there are some characteristics of enzyme that are do not initiate reaction, accelerate the rate of reaction. Enzymes are highly reaction specific, do not alter the equilibrium constant of reaction, remain unchanged chemically after the reaction and needed in a very small quantity, and activity can be regulated by activation or inhibition as per cellular need. So there are also some definitions are very important when we are going to discuss about the enzymes, and these definitions also important for written and viva examinations. So simple enzyme, how do we define the simple enzyme? This is the only protein part of the enzyme, and hollow enzyme or complex enzyme are the protein part of enzyme which is known as apoenzyme plus cofactor or coenzyme. Cofactor can be defined as these are the trace elements or inorganic metallic ion that helps in the enzymatic activity, such as iron for the catalase peroxidase, zinc for the alkaline phosphatase. And coenzymes are the relatively heat stable organic low molecular weight non-protein organic substances tightly bound to enzyme and helps in the enzymatic activity. Mainly the vitamin B complexes act as mainly the water soluble vitamins usually act as the coenzymes that helps in the enzymatic activity. There are also some non-B complex such as coenzyme Q, biopterin also act as coenzyme. So these are the coenzymes. Here I am showing you some coenzyme, that is the vitamin B1, which chemical name is thiamine, or coenzyme form is known as thiamine pyrophosphate, or active form of vitamin B1 is known as thiamine pyrophosphate. So another vitamin B complex is vitamin B2, which chemical name is riboflavin, and active form or coenzyme form is known as flavin mononucleotide or flavin adenine dinucleotide. Vitamin B3, which is the chemical name of vitamin B3 is niacin, and a coenzyme form is known as nicotinamide adenine dinucleotide or nicotinamide adenine dinucleotide phosphate. And this pantothenic acid also known as coenzyme Q. Vitamin B6, which active form or coenzyme form are pyridoxal 5 phosphate, pyridoxal 5 prime phosphate, and pyridoxamine. Biotin, which coenzyme form is also known as biotin. Folate coenzyme form is known as tetrahydrofolate, and vitamin B12 coenzyme form is known as methylcobalamin. Active site, small area of enzyme which binds substrate for catalysis. Allosteric site is the specific site on enzyme other than the active site. There are some substances that bind into the allosteric site and they regulate the enzyme function. That's why the allosteric site is also very important. And substrate is the reactant of a biochemical reaction is known as substrate. Mechanism of enzyme action, that means how enzyme acts. As we know that there is a site on enzyme which is known as active site, where the actually substrate is substrate binds. So at first substrate enters the active site of enzyme and form a enzyme substrate complex. Then the substance is converted into product, and the product that leaves the active site of the enzyme. This is the all over mechanism of enzyme. So enzyme substrate complex formation follow two model or follow two laws. This is known as lock and key model, and another one is induced fit model. So lock and key model, here is the active site is rigid and only substrate with matching shape can fit, and it is the older model. That means in this case enzyme is very much specific, enzyme must have a specific substrate to bind with. For example, glucokinase, this enzyme can only bind with glucose, not other hexose. Okay, and induced fit model, here active site is flexible, greater range of substrate specificity, shape of enzyme substrate and active site adjusts to minimize it. Suppose hexokinase, hexokinase is an enzyme can bind with any other hexose like glucose, fructose and other hexose. So, this hexokinase enzyme follow the induced fit model for the enzyme substrate complex. So, that is all for today. The next day I will discuss about the factors affecting the enzyme activity. Thank you.